Hello, everyone, and welcome to today's live broadcast, Advances in Sedimentation Analysis. I am Dr. Chad Schwartz, Senior Application Scientist at Beckman Coulter, and I'll be your moderator for today's event. We are delighted to bring you this educational webinar presented by LabRoots and sponsored by Beckman Coulter. Beckman Coulter is a leader in centrifugation and flow cytometry and has long been an innovator in particle characterization, laboratory automation, and genomics. Beckman Coulter products are used at the forefront of important areas of investigation and discovery. For more information, please visit beckmancoulter.com. We have a few important announcements before we begin. Firstly, there is a blue Files button at the lower left of your screen that will allow you to access a downloadable PDF of the presentation. Secondly, this webcast is designed to be interactive, and we encourage you to ask questions during the live event. You can submit questions by typing them in the Q&A box, which can be found at the lower left of the presentation window. We'll try to answer as many questions as we can. Additionally, there will be a few poll questions that we can ask you to answer near the beginning of the presentation. The answers to these questions will allow Dr. Demeler to gain a better grasp on the audience's experience with analytical ultracentrifugation. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problems through the green Q&A button lower left. I would now like to introduce today's speaker, Borries Demeler. Dr. Demeler started his education at the University of Montana, earning a degree in chemistry and mathematics. He then joined Professor Ken Van Holde at Oregon State University for his graduate studies, earning a PhD in biochemistry and biophysics with his work involving biophysical characterization of chromatin. It was in Dr. Van Holde's laboratory that he first learned about AUC in a 30-year-old Beckman Model E instrument with UV absorbance optics and a strip chart recorder. He digitized the instrument and wrote his first version of Ultrascan to analyze AUC experiments. Next, he moved to Texas to join Professor Jeff Hansen's laboratory at the University of Texas Health Science Center in San Antonio for a postdoctoral position, where he ported Ultrascan to the XLA, XLI instruments. After completing his postdoc, he joined the biochemistry faculty at UTHSC San Antonio and became director for the Center for Analytical Ultracentrifugation of Macromolecular Assemblies, which he still manages today. I am delighted to now turn it over to Dr. Demeler for his presentation.
Hello, everybody. Good morning, and thanks for joining. I want to also thank Dr. Schwartz for introducing me. Today's seminar will be on sedimentation analysis. I will tell you a little bit about what we're doing in our lab in order to improve Ultrascan, a software package for the analysis of AUC data, and tell you what methods we have implemented recently to address specific problems in biology and nanoscience. My talk will cover the following topics. First, I will give you a background. Then I will touch on the parameters that we can pull out of AUC experiments, size, anisotropy, and density, and discuss optimization methods that are implemented in Ultrascan. I will focus on two different classes of analysis methods, one dealing with non-interacting systems and those that deal with interacting systems. And I will present examples to illustrate what we're presenting here. So first, what can we learn from analytical ultracentrifugation? AUC is an excellent method for characterizing any molecule you can put into a solution. You can study their interactions, and you can do it in a solution environment with very, very small amount of sample. You only need about 0.5 ml in order to study it in the AUC. And you can run up to 14 samples at the same time. The analysis is based on first principles, which means that we know mathematical equations to describe everything that goes on inside of the ultracentrifuge cell. And so we don't need any standards like you would need in column chromatography. We also have much higher resolution than, for example, in light scattering. The molecules can be studied in a physiological environment, so if you deal with proteins and DNA and other biomolecules, it's sometimes important to be able to modulate the solution conditions. So you can study the effect of pH, ionic strength, what kind of buffer you're using. You can even study things like oxidation state by adding reductants. You can add ligands, such as drugs, and see what they do to your samples. You can modulate temperature, and you can modulate concentration, and all of these things turn out to be very useful when you study biological samples. Furthermore, by changing the speed with which you run your experiment, you have access to a very large size range of molecules you can study. Anywhere from a few hundred Daltons up to tens or hundreds of millions of Daltons can be studied. Because we're measuring in a solution environment, we also have access to the dynamics of the system. So you can measure oligomerization states of reversible self or heteroassociating systems. You can measure their stoichiometries. You can measure ligand binding, and you can even measure the slow kinetics that may be involved in a reaction, as well as the equilibrium constants. Another application is to study the composition of samples. Maybe you have an unknown sample and you don't really know what the composition is. Then you can get access to the number of components, their partial concentrations, molecular weight, size, and anisotropy. Finally, you can get access to the shape of a molecule, at least indirectly, by doing conformational analyses, such as like folding and melting studies of biopolymers, conformational changes that are induced by changes in the solution properties. Here's a quick review of the experimental setup. You have a rotor that can be spun up to 60,000 RPM, generating up to about 300,000 G, which is a pretty high force, makes even small molecule sediment. The sample is contained in a sample compartment like you see here on the left, which is sector shape. So the flow goes with the sector shape of the compartment. We don't have any wall effects. We have a meniscus at the top of the solution column, and we form a moving boundary during the sedimentation process that you can track by adding light or by scanning the cell with some wavelengths that could be either UV or visible, or using Rayleigh interference optics or fluorescence. The light that gets absorbed by the sample can be measured, captured below by a camera, and you get an observance trace that shows you a meniscus at the top, which is sort of a refractive artifact giving you a spike. And then you see the moving boundary that sediments and diffuses at the same time. And you see the concentration represented as an absorption trace. You make these measurements, these scans, over time. You do multiple scans, generating a data set that looks like this blue one here, where you have multiple traces showing you what the concentration distribution looks like over time. When we start, everything is uniformly distributed throughout the cell. So if red is our analyte here, it is uniformly mixed throughout the cell, and we get a horizontal scan or horizontal line showing uniform observance throughout the cell. But as soon as we spin up the rotor, we form a moving boundary, and you see depletion near the meniscus where there's no longer any absorbance, and then you see this moving boundary which also diffuses. This whole process is described by the Lamb equation, which you see here up on top. And this is a second-order partial differential equation, which you can solve using finite element. And the thing that I want to just point out here is that you have a sedimentation term describing the sedimentation transport inside of the cell, circled in blue, and you have a diffusion term, which describes how the boundary that you create diffuses over time to equilibrate that concentration gradient. If you go to the end of the experiment, you have an equilibrium condition in which the sedimentation and diffusion terms cancel out, and no more net flow occurs. And when you solve this differential equation, all the time terms drop out, and you end up with an exponential, and you see an exponential trace. The slope of the exponent, the slope of this exponential shape is then proportional to the molecular weight. So when we fit the Lamb equation to our experimental data, we essentially get two pieces of information. One is the sedimentation coefficient, and the other is the diffusion coefficient. And we get that for each analyte that's in the mixture. These parameters, once we have obtained them, can be used to obtain additional details about the sample. So for example, we can take the diffusion coefficient and plug it into this equation to get the frictional coefficient of our sample. Similarly, the S value that we get, the sedimentation coefficient, together with the frictional coefficient, can be used in this equation to obtain the molecular weight. This also requires knowledge of the partial specific volume, and it requires knowledge of the density of the solvent in which we're measuring. Once we have the molecular weight, together with the partial specific volume, we can also obtain the volume of the molecule. Once we have the volume of the molecule, we can determine the radius of an equivalent sphere. We call that R0. If we plug the R0 into the Stokes-Einstein relationship, the second to the last equation shown here, you can obtain the frictional coefficient of the equivalent sphere. Together, the frictional coefficient obtained in the first equation and the equivalent sphere frictional coefficient make up the frictional ratio, which gives us the anisotropy, which I denote by phi. This will give us an idea about the globularity of the sample and tell us something about the shape. So what we get then is we get some information about the shape of the molecule, we get some information about the size, and we get some idea about the anisotropy of the particle, which is also related to the shape. And then there's this density dependence over here, where we have information about the partial specific volume of the molecule and the density. Now, please note that we can only get two parameters out at any given time. The third one needs to be supplied, and ordinarily, this is our partial specific volume. The density of the solvent can be measured independently and provided through third methods. Now, in a non-interacting system, such as this one, you can see we have multiple boundaries. We add the Lamb equations together and multiply each Lamb equation for each component that's in the system by some amplitude Ci. Okay? So the Ci then represents the partial concentration of our solute. In an interacting system, the situation is a little different. And here the sedimentation diffusion coefficient is not constant throughout the experiment, but it is dependent on the mass action equation of whatever reaction is occurring. So for example, if you have a monomer-dimer equilibrium as shown here, we can get the association constant by dividing the concentration of the dimer by the concentration of the monomer squared. Now, the ratio of dimer versus monomer is dependent on the concentration inside of that moving boundary gradient that we have. So at the top of the gradient, we have higher concentration, so we get more dimer, and at the bottom, the dimer will fall apart into monomer at low concentration and be mostly monomer. So what we're measuring then is the weight average sedimentation coefficient shown here and the gradient average diffusion coefficient for all species that are in the mixture. And so we just modify our Lamb equation to fit the gradient average diffusion and the weight average sedimentation coefficient in a case like that. And I will then show you methods for how we can access this information for each one of those systems. So we have a velocity experiment, and we can ask questions such as, how many components are in there? What are their sizes and their molecular weights? What are their anisotropies? What are their partial concentrations? And do these components interact? And if so, how fast and how strongly do they interact? And then finally, we want to know what's the reliability of our measurements. So we want to get some statistics. We have several methods for analyzing the data that comes back from the analytical ultracentrifuge. These are all, so let me just go through them. One of our basic methods is the two-dimensional spectrum analysis, or 2DSA, which gives us a high-resolution, general, and model-independent solution for both size and anisotropy distributions of non-interacting systems. Next, there's the parametrically constrained spectrum analysis, or PCSA, which is similar in the way to the 2DSA in that it identifies size and anisotropy relationships, but here for polymerizing systems, and it provides a constraint fit over the two-dimensional sedimentation diffusion space. There's also custom grid analysis, where we can take advantage of prior knowledge that we may have from another experiment. We use that to constrain our data set and parameterize the 2DSA grid in terms of alternate hydrodynamic variables. And we have the genetic algorithms, which is an optimization method, which will give us robust nonlinear least squares optimization, and it provides a parsimonious regularization of the 2DSA spectra. What that means in detail, I will go over in a minute. This is also used for fitting of discrete nonlinear models such as reversible associations, non-ideality, co-sedimenting solvents, and other complicated systems. Then we have a Monte Carlo analysis, which allows us to extract the statistics of the parameter distributions that we measure and allow us to assign things like how much a sample's parameters are distributed. And then we have the Van Holt-Reichert analysis, which gives us very detailed sedimentation, diffusion-corrected sedimentation profiles, which allow us then to compare samples, let's say in different conditions, different ionic strengths, different pH, and get even slight differences with great resolution. There are also other methods such as the CFS and the CFS, F/F0 and so forth that are implemented in other software packages. These are rather low resolution, and we generally don't use them in Ultrascan. So here's an illustration of the problem that we're trying to solve then. If you have a mixture of different sizes and shapes in a solution, then what you would like to know is, where are they? So if you have a two-dimensional space here, which maps out the shape, let's say, on the y-axis and the size on the x-axis, you could imagine that you have a molecule that's very globular with a frictional ratio close to 1 and a small size in the lower left here at that blue dot. And the shape of that curve would look like the pattern that you see above, which is very closely spaced boundaries because it's small, it sediments slowly, but it spreads out very rapidly. It has large diffusion because it's very globular. In contrast to that, you can have something that is very elongated and very large, which would sediment very rapidly because of the large size, and so the boundaries would be spaced far apart, and you would get steep boundaries because of the elongated shape. The diffusion would be very slow, and so you would see something like in the upper right. And then you can have intermediate cases in between. And then the question is, how much of each of these components do you have? So we would then, for each dot that we find in our mixture, representing one solute or one analyte, we would then need to know what the partial concentration of that is. So after we do our analysis, we end up with a picture that sort of looks like this, where we can represent the partial concentration of each analyte that we find by a color density. So a faint color would represent low concentration, and an intense color would represent high concentration. This can also be shown in a three-dimensional plot like you see here, where the height of the peak represents the concentration. Now you will also see me show distributions from the Van Holt-Reichert analysis, which can be either represented in cyan as integral distributions, where you can read off the y-axis the relative proportion in percentage, and from the x-axis you get the amount or the size or the sedimentation coefficient at which the relative amount is sedimenting. So here we see like 4 or 5 different components that sediment at different speeds and have different relative amounts. You can also show this as a differential distribution shown in green, which looks more like a chromatogram you might be used to. But the nice thing about the integral distribution is that you can go immediately to the y-axis and read off the amount of material that might be there. So a quick explanation of the two-dimensional spectrum analysis. You simply take this two-dimensional grid that we have, and you solve the Lamb equation for each one of those grid points. So each grid point, of course, represents a different sedimentation and diffusion pair. And so we solve it for all of them. And knowing the frictional coefficient at each point, we can plug that in together with the sedimentation coefficient to get the diffusion coefficient. So we have a diffusion coefficient and a sedimentation coefficient that we can plug into our Lamb equation to simulate. And now our question is simply, how much of each component do we need, which is shown down here in the sum? And then we subtract it from our experimental data, and we're trying to make that difference a minimum. So that's called least squares fit. And if we have satisfied this minimization, then the amplitudes for each individual component will tell us how much of each species that's in our mixture is there. We use a process called NNLS, or non-negatively constrained least squares, which guarantees that these amplitudes are either positive or zero. Zero if the sample or the analyte isn't present, then the amplitude would simply be zero. And if it is present, it will be a little bit positive. So to get high resolution, we move this grid around until we have whatever desired resolution we need, and we save the positive contributions in the storage grid here, which then becomes our answer. This can be done in parallel. You simply take each grid moving and put it into a different processor and calculate them all at the same time, and you collect the results into a final grid, which will give you the answer. In the next step, you might refine the solution. So here you see a picture of what you might get back from the 2DSA, and you draw a box around each solute that was found, and then you use the genetic algorithms to evolve a solution that will then also filter out solutes that may not be there or may actually be just noise and false positives. So this is called parsimonious regularization, and it's achieved by the genetic algorithm. And all of this can be done on a supercomputer. This is the genetic algorithm solution that you might get from the previous 2DSA distribution. So you can see we end up with much fewer components. And this solution satisfies Occam's razor, meaning the simplest solution that gives you the same RMSD or quality of fit is the best solution, is the one that we really want. So we filtered out all the noise and the noise contributions and these false positives, and we got rid of them by genetic algorithms, which figured out which are the salient components that we really have in the system. And again, this is done on a supercomputer. All of this is done with the Ultrascan software, using the Exceed environment, which is an NSF-funded supercomputing environment, or called Extreme Science and Engineering Discovery Environment. And we have this Ultrascan Science Gateway, which is supported by NSF, which allows each institution that has an analytical ultracentrifuge to have their own instance and then run these analyses on the Exceed infrastructure. And it's all free. So if you have an analytical ultracentrifuge and want to use it, talk to me and we'll set you up with one of those instances, and you can use that for high throughput and high resolution analysis. There are several supercomputers across the United States that we use, and we also have an implementation in Germany that serves Europe and the Middle East. And so this is distributed and in production use pretty much worldwide. So let's look at another method here, the parametrically constrained spectrum analysis. So this is a very simple system. We just mix two analytes together. In this case, we have a globular protein of about 14.3 kilodalton lysozyme, and we mix it with a 208 base pair DNA fragment, which is a extended linear double-stranded DNA fragment. And the question we would ask then is, can we get a good description of this heterogeneity in size and shape using an analysis method that is fairly simple to do. So one of the low-resolution methods that exist that would give us a univalued parametrization, meaning one S value for one shape, is the CFS. It basically uses a horizontal line to represent the sedimentation space and then moves that line up and down in the frictional ratio domain to find the best possible fit. So if we have two different anisotropies for DNA and protein here, then what we would have is a weight average frictional ratio that would then represent both of these, but it wouldn't really represent the true frictional ratio of each species. If you use the two-dimensional spectrum analysis, you see this result shown here on the left. You can clearly see that the different anisotropies are well represented by this approach, but you still get quite a few signals returned due to the uncertainty of the measurement. And but much better than with the CFS, you get the true anisotropy represented. This is the same picture shown in the differential format. So if we use a genetic algorithm analysis, we can then do this parsimonious regularization and really pin it down on the two actual solutes that are really there. This is really nice, but unfortunately it's also computationally expensive, and it takes quite a bit of time, and you need to run through this whole refinement process. So we're wondering if there's a better way to do that. And so what if one, instead of using the CFS analysis, which uses a horizontal line, simply used a sloped line and did the representation of all the sedimentation coefficients that are possible on a sloped line that actually goes right through those two solutes. That would give us a general method that provides a univalued relationship between size and shape. And this was published last year. If you're interested, you can look it up, how it's done exactly. But let me just show you some of the results here or how this approach works. So this PCSA approach then selects any single valued function, like a straight line, hyperbolic functions, increasing, decreasing sigmoids, exponential growth. You pretty much can do whatever you want as long as it's single valued, and then generate a discrete grid of these functions by varying the function's parameters. For example, you might want to change the slope of the line. You might want to change the intercept of the line to cover the entire space. And each line would be discretized across sedimentation and diffusion, the two-dimensional parameter space, and optimized with NNLS. So here are some examples of the functions you could use to cover the entire two-dimensional space of S and D, or size and shape. So in the upper left, you see a straight line parameterization. On the upper right, you see an increasing sigmoid. On the lower center, you see a decreasing sigmoid. If we fit this particular sample of lysozyme and DNA with a PCSA, we get this result. And so here you see all the different lines shown in different colors, and the colors represent the quality of the fit. So every time we check a different representation of that line, we get a different RMSD, or quality of the fit. And we assign a color to it that ranges from very low, meaning a very good fit, in red, to purple, meaning a very poor fit. So you can see that this diagonal that goes from the lower left to the upper right represents a good fit. And there's one line that gives the best fit, and that's the one that we pull out. And if we compare that to the genetic algorithm result, we see that they're pretty much right on top of each other, giving us the same result. So that's very encouraging. So now we apply that to represent a polymerizing system. In this case, simply a restriction digest of DNA. In this case, you have many different sizes that are different molecular weight, but they're also different shape, because double-stranded DNA, when it's linear, becomes more extended the larger it gets. So we want to capture this relationship between size and shape by modeling it with a PCSA along a single line. And here we use an increasing sigmoid, and as you can see, we have perfect correspondence between electrophoresis gel and the 2DSA pattern that we get back. So if you have very high concentration analytes, like the two bands in the middle, we'll get high intense peaks, representing the concentration and the size and the shape. If we pick one of these fragments, or two of these fragments, and look at them individually and run them with PCSA, again, we can use a straight line to represent those two components, and we get this picture. Now, this is a fragment that is 208 base pairs for the lower one and 2811 for the upper one. So the upper one is very extended, giving us a frictional ratio of about 7, and the lower one about 3.5, something like that, in this particular salt concentration. So if you were to do the same thing with the genetic algorithm, you would get that red distribution shown in the upper right. And again, it looks very similar to what we get from the PCSA, which is shown here in green. Now below it, you see the CFS result, and I want to point out why we don't like to use CFS. So in this case, for the lower species, you would get many different species distributed around the peak that should really be a single peak, but now it becomes a broad distribution. And only the upper one is actually shown as a single peak. And so that's a problem. So if we look at that, we see that instead of getting the single peak shown in blue in panel E here, you get many different peaks that really don't represent this species at all. And the reason for that is in the least squares fit, where we basically force a weight average frictional ratio that is too high on top of this more largely diffusing sample. And that causes all these different small amounts of false positives to be put into the solution, giving us this broad spectrum. In the middle, you see a line that shows the same solution, but using maximum entropy regularization. And below it, you see the Monte Carlo analysis. So regardless what approach you use, it still gives you a very broad pattern. And if you look at it in a differential profile, you see many different species popping up when they in fact should only be one, like in the PCSA analysis. So if you transform this result into a molecular weight domain, you will again see that both of the values are going to be off. In red, shown the C of S, while in the PCSA analysis, you get very close to the target. The target is shown in green, and you see that even the PCSA is a little bit off, but it's systematically off, meaning both sites are both solutes are a little bit off, and that is because we had the incorrect V bar used for this analysis. So we should have adjusted it so everything would have moved down a little bit, and then it would have hit the target right on. So the PCSA can get you actually the correct molecular weight, while in the CFS you might be stuck with a misleading result. Here's another example. This one shows how we can use the PCSA analysis to study a process like clathrin assembly. Clathrin is this molecule that has these three arms called trischelia, and they interdigitate to form these baskets, and this is important in exocytosis in the cell. So these baskets essentially shuttle molecules out, in and out of the cell. And this clathrin assembly then goes from a very non-globular molecule, which basically drags through the solution like an anchor, to something that is spherical and perfectly globular. So we want to then see if we can model this process of going from non-globular to globular and see the change in shape and size simultaneously with analytical ultracentrifugation. So when we did the AUC experiment and analyzed it with 2DSA, we get lots of different peaks here showing many different components in there. And we also see that at low molecular weight or low size, we get very high anisotropic values representing that non-globular shape of the monomer. But as soon as the monomer assembles even into very small cages, we very quickly get to a distribution of fully formed cages, and they're spherical, having a frictional ratio of 1. And then you can see in the lower right of this panel the size distribution of the cages all along the one-axis of the frictional ratio. So, but again, we have lots of different peaks here, and it's not quite clear which ones are false positives and which are real. So if we force the solution onto a PCSA grid, trying to find a univalued relationship between size and shape, we can use the decreasing sigmoid, which then covers the distribution of anisotropic monomers going to assembled Klassen cages being more globular as the molecule grows bigger, which is sort of the opposite of what we've seen in the DNA case. In this case, we get a nice clean picture that shows you the monomer here and maybe some dimer, and then the dimer, trimer, and so forth. They assemble very quickly into fully formed cages, and the picture becomes much more globular for the larger species. So this is a nice representation of what might be going on in the assembly reaction, and makes a little bit more sense than the 2D assay. Here is just a picture of how well the PCSA solution actually fits the experimental data. In red is our solution overlaid on top of the black experimental data, and we get a perfect trace showing very good fit. Sticking with the Klassen theme for a moment, this work was done, by the way, in collaboration with Eileen Lefer in our department, and she's also interested in how these Klassen molecules assemble to form cages. And this is assisted by intrinsically disordered protein called AP180, or auxiliary protein 180 kilodalton long. And it has all these binding sites that captures the terminal domain of the Klassen molecule and then brings them into sufficient proximity so they can form cages. So we were looking at this interaction between these binding sites in that AP180 and the terminal domain of this Klassen molecule. So we took a small fragment that contained one binding site and we labeled it with a fluorophore. And we were using then the fluorescence optics to study this association by following the fluorophore. Let me give you an example of how you can see that. Now here we have a Van Holt-Reichert distribution that starts off in black with just the fluorophore labeled a small 5 kilodalton fragment without any Klassen added. Then in purple, blue, and cyan, we add increasing amounts of the Klassen domain, which by itself sediments like the red line. This was measured in UV because it wasn't labeled, so we had to look at it in UV. And you can see that we form a complex that's slightly bigger than the Klassen by itself. So then we can titrate more and more TD40, or terminal domain, which is about 40 kilodalton, into our mixture and form more and more complex. And by following that complex formation, we get the isotherm of binding, and we can derive the KD for this reaction. So here is just one of the concentrations, concentration ratios, and you can see we have some free Alexa sedimenting near zero in the molecular weight. Then we have that free fluorescently labeled molecule, and at 45 we can see our complex. Now the free TD40 is not visible because it's not labeled in this experiment. So all we see is the free and the bound, but by knowing how much we put in, we can fill out our KD equation and calculate a KD for this reaction. And by following the assembly reaction then over a range of different titration mixtures, we can then fill out this isotherm and get a value for the association. And in this case, we compared it to NMR experiments, and there's a single binding site and a second binding site species as well measured, and you can see the differences in binding strengths here. And it gives very similar results as what you measure in an NMR experiment. The next example is an example of a self-associating system where you have two proteins that are identical coming together forming a complex or a dimer. So this is the monomer-dimer equilibrium. In this case, this was work done together with Adrian Coutinho-Clay from UT Austin. And he's studying these multi-modular polyketide synthase molecules. And they have this dimerization interface, which he crystallized. And so we wanted to study the KD for this association. We did that by running the analytical ultracentrifuge with simply that polyketide synthase at different concentrations. And what you see are these sort of half polynomial Van Holt-Weisheit distributions that it increasingly shifted to the right as you increase the concentration of the molecule. And the reason for the shift is because mass action determines that at higher concentration you form more dimer. Well, the dimer sediments differently than the monomer, and you get this weight average shift of the sedimentation profile over to the right, and the closer you get to complete dimer formation, the closer you get a complete sedimentation profile just for dimer. So again, by looking at the different concentrations and looking at the weight averages, we can then fill out a binding isotherm from which we can derive the KD for the monomer-dimer reaction. Now that's one way to do it. Another way to do it is to fit the Lamb equation directly with the weight average sedimentation coefficient and the gradient average diffusion coefficient at each point in the boundary for all the scans. And again, after you verify that you have a shift in the sedimentation coefficient as you go to higher concentration, and here you see a Van Holt-Weisheit distribution in the lower left for a low and a higher concentration, you can clearly see the shift. So then you know that you have a monomer-dimer equilibrium, and you might want to fit it, and then you get the exact values for the KD directly from a single experiment fitted with a genetic algorithm analysis. This is a Monte Carlo analysis of that. And so you also get standard deviations for each parameter that you fit it. Finally, I want to mention a few words about the partial specific volume. That's sort of a stepchild of analytical ultracentrifugation, because most of the time people just estimate that value, or they take a literature published value. But it turns out the V-bar is very sensitive to solution conditions, different salt conditions, and so forth. So oftentimes the V-bar values are actually wrong, and so the molecular weights that you calculate are then off as well. And you've seen that in that DNA sample that I showed, where the PCSA molecular weights were a little bit off target, and the reason was an incorrect V-bar that wasn't correctly measured. So how can we get to this value? Well, there's a very simple way, but it requires a lot of sample, and it needs to be very homogeneous, and that's in densitometer. You just measure it in a densitometer. So, but that's oftentimes out of the question because you don't have enough material. Another way of doing it would be to do density matching experiments by sedimenting the sample simply in different ratios of heavy and light water. And by increasing the density of the solvent, you change the buoyancy term as a function of density, and you can simply look for the point or extrapolate to the point where there is no sedimentation at all, and that would be equivalent to the density of your macromolecule, which you can then take the inverse of to get the V-bar. So that's another way of doing it. And there was a paper published recently from our lab that, together with Craig Peterson, that implemented this approach. Another way of doing it would be to use the custom grid approach. This is the last method I'm talking about. And this one allows you to take prior knowledge from other experiments that you might have to constrain the solution to other parameters. And finally, there is a way to do it with using analytical buoyant density gradient centrifugation, but that's very limited, and I'm not going to talk about that today. So I will now focus on the custom grid analysis and show you how that works. This was published last year in this paper here. And here we use prior knowledge from other experiments that we can introduce in the AUC fit as a constraint to get access to the other values, such as the V-bar, that we normally wouldn't get. So for example, if from densitometry or from sequence or density matching you do have the V-bar, you can fix that and then obtain molecular weight and anisotropy like I've shown previously. But if you have electron microscopy data, or small angle X-ray scattering data, or X-ray NMR structures, you can fix the anisotropy and obtain the V-bar and molecular weight. Now that's only true if all of the molecules in your mixture have the same anisotropy. Then you can use this approach. Now that's simple if you have a single species, but if you have heterogeneity, you need to make sure that all particles fit that bill. But sometimes you also have the molecular weight, either from mass spectrometry or X-ray crystallography or polymer sequence. In that case, you can fix the molecular weight and determine the V-bar and the anisotropy. So I'll give you one example here. A couple of years ago, this paper came out, and it described an ultra-stable silver nanoparticle. And these people crystallized this particle, so we know actually what the structure of this particle looks like. And as you can see, there is a silver core that is then, using Mercator linkages, linked over the sulfur atom to these benzoic acid residues. And these benzoic acid residues have the carboxyl group and the hydroxyl group here on the outside, as it's shown in this picture. So inside we have the metal core, then comes a hydrophobic shell from the benzene residues, and then on the outside you got the carboxyls and the hydroxyls, making it hydrophilic. When we ran that in the analytical ultracentrifuge, we saw a single species corresponding to the monomer of this crystal here, of this nanoparticle. So there is another feature in Ultrascan, which is called bead modeling. It allows you to take a crystal structure and determine from it the frictional properties of this molecule as well as the volume. So just using the crystal structure, which does not contain any waters, we can then calculate the partial specific volume of this, or estimate it, for the anhydrous particle. And we determined that to be 0.24 mils per gram. The frictional ratio was determined to be 1.25, fairly globular, not an exact sphere, and you can see why. There are these things sticking out, these benzoic acid residues sticking out, giving it some frictional surface, which then increased the frictional ratio slightly above 1. We measured the diffusion and the sedimentation coefficient. Now, from the AUC experiment, we can fix the molecular weight because we know exactly what that is from the crystal structure. But what we don't know is the partial specific volume, because that includes the hydration. So since this didn't include any waters, we would expect our partial specific volume to be off. But using the molecular weight, which we have, we can then fit the partial specific volume and the frictional ratio. As you can see in the lower left here, there is a single species, and from that we can derive the frictional ratio and the partial specific volume. Now let's compare these numbers. So from the AUC experiment, we get a frictional ratio of 1.47. From the prediction, we only get 1.25, which is slightly lower. So why would this be less than in the observed experiment? Well, look at where these carboxyl groups, or the hydrophilic groups, are. This is where the water molecules will bind. And this is at the end of these spikes coming out from that silver nanoparticle. So that would make it even spikier, or more extended, or more frictional. That's where that difference comes from. So it would be the bound waters that actually add to the frictional elements. In addition, we have a different partial specific volume, which is slightly higher than the simulated one, or the calculated one from the X-ray structure. So why is our partial specific volume higher? That would imply a lower density. Well, the waters that are bound to the sedimenting particle have a much lower density than the silver metal. So as we add the water molecules, we actually decrease the density of the overall particle. So now we have a way of estimating how much water is actually bound to this molecule by simply measuring the difference in partial specific volume between the calculated anhydrous version and the one observed in an AUC experiment. So this sort of wraps up what I was trying to convey today, which is the different things that you can learn from an AUC experiment and how you can analyze it using our software, the Ultrascan software, to derive these values. And I want to thank you very much for your attention, and also point out that other people have contributed to this research, and credit them on this slide. And you all have a PDF version which you can download. In the interest of time, I'm going to skip over acknowledging each person individually here and just go on to our last slide here, where I would encourage you to send me any email with your questions. We have a facility here in San Antonio at the Health Science Center where we perform AUC analysis as a service, and we can design custom experiments for your needs. We can analyze the data. We can design experiments for you. And we also provide high-performance and high-throughput analysis solutions for AUC experiments with our software. So if you need that, help with that, let us know. And we will help you with this. And we also provide training for AUC. If you have a graduate student or want to learn it yourself, feel free to contact us, and we'll set up a workshop for you. These are the websites. You might want to visit us on the web. And thanks again, and I'd be happy to answer any questions.
Great. Thank you for that highly informative presentation, Dr. Demeler. You've successfully demonstrated the versatility of Beckman Coulter's AUC platform. Before we get started on the question and answer session, I would like to ask a few poll questions to gauge the user experience of AUC. So you should see a window pop up in front of you. The first question is, do you have an analytical ultracentrifuge in your lab today? Pretty simple question, yes or no. We'll wait for the answers to populate here. I'd also like to mention, please stay with us. We will get to the question and answer session immediately after these poll questions. It looks close to a 50-50 split. We're at 54% no and 46% yes. So we have a very knowledgeable user base here, a very strong audience, it seems. We'll get to our next question as well. The next question is, for which applications do you use or would you use your AUC? There's multiple answers are allowed here. Molecular weight, heterogeneity or molecular aggregation, interacting systems, as Dr. Demeler talked about today, molecular conformation, stoichiometry, or other. Again, we'll wait a few seconds to let the answers populate. All right, it looks like the majority of folks use AUC to study heterogeneity or molecular aggregation. That's a hot topic these days. Molecular weight is also up there at 24.1%. Interacting systems at 17%. Molecular conformation, 16%. And stoichiometry, 12.8%. That's we see a lot of your basic research with AUC. Other comes in at 4.5%. We'll get to our third and final question, and then we'll have some questions that you guys have posed as well. The third question is, what types of particles do you or your lab typically study by AUC? Again, multiple answers will apply, so just go ahead and check whichever boxes you use. Biopolymers, synthetic polymers, viruses, nanoparticles, or you're not a current user but you're exploring the possibility of AUC. The question should populate shortly. In the interim, I would like to remind our audience to submit questions by typing them in the Q&A box found at the lower left of the presentation window. All right. Wow, a vast majority is biopolymers, 46.2%. That will consist of your
Okay, this is Don. We seem to have lost Chad. I'm going to go ahead and pose the questions that we have from the audience. We'll start off with the first question from Julia. We'd like to explore the AUC to characterize a virus drug product. However, there is a lot of literature found about viruses. We'd like to see residual host proteins as well as the three forms of the virus. Is this possible with AUC? What would be the best strategy as a start?
Okay, that's an excellent question, Julia. let me try to address this. You're right that there's not a lot. Can you hear me?
Hi, Dr. Demeler. I apologize. We got disconnected here. I don't know if you went to the next question, but feel free to moderate. I apologize again.
Okay, I hope everybody can hear me. So, Julia, to get back to your excellent question, how to characterize drug binding to a virus. So, in an analytical ultracentrifuge, you can observe the hydrodynamic behavior. So, if you have something binding to a virus particle, you would need to see a hydrodynamic change. Now, this can be also detected indirectly. Let's say the drug causes some sort of aggregation. It would be easy because you would see the aggregate forming as soon as you add the drug. But let's say you have a huge nanoparticle or a huge virus particle, and you bind the drug to it, and you don't really see much of a hydrodynamic change because the size difference of the complex versus the free virus is very, very small. So there's not much of a hydrodynamic signal. But let's say your drug was had a unique chromophore that is not present in the virus. So a virus is made out of protein and RNA, protein and DNA, so it would absorb in the UV but not in the visible. If you could attach a tag to your drug that would make it visible in the visible range, maybe a fluorophore, you could then just follow the drug. And when the drug binds, it would get a huge difference in sedimentation, which you could easily see. So that would be a way to do it. There would be one way of doing it. It really depends on the individual case that you're trying to do, that you're trying to analyze, of how you would design the experiment. If you want to catch me offline later on, I'd be happy to discuss it further with you. So now we have another question here. I'm not really sure where they are, but Chad, do you want to read the next question?
Yeah, I would be happy to. The next question comes from Francesca Matrioli, and the question is, in PCSA analysis, how do I choose the linear versus sigmoidal lines? Should I always test both?
Okay, yeah, that's easy to answer. So in Ultrascan, if you run this on the desktop, you have a drop-down box where you can select which one you want to use. On the supercomputer, if you use the LIMS interface, you can actually tell it to just test all of the methods and just let the computer figure out which one is the best one. In the supercomputer, we can, do a very quick analysis of this and run this all in parallel, so we just run all of them at the same time, and then the best one gets returned. So there you also have a drop-down box where you can make an individual selection, and one of the options is to run them all, and so that's what I would recommend you do.
Great, thank you for that. Our next question is, what is the best way to report results from grid analysis? How do you interpret and or deal with overlapping or smeary peaks that remain after grid analysis? This is important when reporting the data, as the size of the box drawn will influence the error, percentage of species, etc.
Okay, also excellent question. So what you have is actually the genetic algorithm, which is supposed to do the parsimonious regularization, meaning getting rid of points that aren't really relevant. So when you end up with a genetic algorithm analysis, you should have just single species. However, the position of these species in the two-dimensional grid depends on noise. So when you're fitting noisy data, you will get a slightly different answer every time you make another measurement because of the noise. The noise affects the output. So you really want to probe what the effect of noise is on the data. And for that, you have to run a Monte Carlo analysis on top of it. So what I would do after finishing with the GA, I would use the GA to initialize a Monte Carlo GA and run that on top of it. And that will show you how much these points move around as a function of the noise that's in your data. And then what you would do is you would integrate each peak that you get back to get an idea about the standard deviation of all the parameters that have moved around inside of that peak. So again, I can show this in more detail. If you want to contact me offline, I'd be happy to go over that with you. But that's certainly possible, and that will allow you to distinguish true peaks and peaks that don't really belong together. All right,
Great.
Next question.
Thank you. We've had a couple questions come in about AUC versus size exclusion chromatography, or SAXS, especially for protein molecular weight analysis. Can you give us a little bit of insight there?
Sure. That's a very important question. So many people try to estimate the oligomerization state by just running secmoles. The nice thing about secmoles is, you separate your protein, presumably the monomer from the dimer, and you then see a monomer peak and a dimer peak. And as it goes through the detector, using moles, you can estimate the molecular weight. However, the problem is that whenever you run something through a column and you calculate the exclusion, you calculate the time it takes for that peak to run off, you can then estimate based on the runtime how big the molecule is. Unfortunately, this totally depends on the shape of the molecule. So unless you have a standard that has the same anisotropy and the same interaction with the column matrix as the protein of interest, you will get a misleading result. Again, you need standards here to make it work. If you use the analytical ultracentrifuge, on the other hand, you're not dependent on standards. Here, the results are absolute. If you have the correct partial specific volume, and I showed some methods on how you can get that, you will get the absolute molecular weight simply from running an AUC experiment. You don't have to worry about whether you use the right standard or not. So, in addition, you get much higher resolution from AUC. It's incomparable to light scattering or column chromatography, just measuring elution times. So it is a really big difference in resolution for AUC. And if you really need to know for sure, AUC is definitely the gold standard, and you don't want to rely on your column chromatography result, because that's just too imprecise. And light scattering by itself will only give you a weight average molecular weight, and this will actually be a very broad peak, which will not allow you to separate monomers and dimers or get KDs. You can only do that in an AUC experiment. So oftentimes when you have a reversible system, you see both monomer, dimer, maybe even tetramer, all in one mixture. And so in order to pull that apart, you really need to use the high centrifugal force available in the ultracentrifuge to actually find out what exactly you have. You will never see that in a light scattering experiment. Okay, next question.
Great, thank you. We are running a little bit short on time, and I want to mention any unanswered questions will be documented and will be answered by Dr. Demeler in text form following the presentation and posted to beckmanculture.com alongside the recorded presentation. We do have time for one more, and I'm going to summarize a couple questions that came in here, but you presented several different types of analysis, 2D-SA, Monte Carlo, PCSA. How do we know, how does a user know which technique or model to really use, and how to trust that data?
A very important criterion for which one you want to use is what it is that you want to learn. And then finally, after you've done the fit, you want to make sure you get a low RMSD, meaning a good fit. So if you don't get a good fit, you can't trust the result. And there are many reasons why you might not get a good fit, including non-ideality, or because your model just doesn't match what you're trying to measure, or because you have some sort of a gradient forming underneath your analyte that causes nonlinear sedimentation, and your model just doesn't account for this. So you need to make sure that you really apply every tool in the toolbox to figure out what exactly is going on with your data. And each method has its own advantages and disadvantages. So which one you use really depends on the case or the data set that you're trying to analyze. And it does come with experience to figure out which one is the best way to do it. But we always start with a 2DSA analysis. And in fact, on our website, there is a guide that will sort of walk you through and tell you what you can learn from each and what is the proper sequence for refining your data set. And generally, you always do a 2DSA followed by a GA, unless you have very broad distributions in which individual components cannot really be identified, in which case you just stick with the 2DSA. But we also pretty much always look at the data with the Van Holt-Weisheit analysis to get diffusion-corrected sedimentation distributions that we can compare. Very fine-grained detail can be obtained that way. And then PCSA, custom grid, those are specialized applications for, let's say, polymerizing systems or cases where you want to know the partial specific volume, like some nanoparticle applications that I showed. But there are many, many different cases where you could use either one of them and probably learn the same thing. So again, if you have some specific examples that you want me to discuss, I'd be happy to talk to you offline and go over that and help you with that.
Thank you very much. Unfortunately, we are out of time. I want to thank the audience for their interesting questions and participation in today's event. I would also like to thank our sponsor, Beckman Coulter, and of course, Dr. Borries Demeler for making today's educational webinar possible. Today's webcast will be available for on-demand viewing through both beckmancoulter.com and labroots.com. We are also excited to offer a PDF version of Dr. Demeler's presentation on the Beckman Coulter website. By registering for this event, you will receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks again. Goodbye.